she got into science, and she mentioned that it wasn't like, I'm sure she probably was running around outside of the stream and in the field and things, but she said that the gateway to actually getting involved in this sort of work was actually art, which, given our recent interest in bringing together art and systems sustainability here at Utah State, it's pretty interesting because somebody actually said, hey, art got me into science. And I asked, well, how is that? And she mentioned, when you think about drawing multiple things, you think about how you actually frame up a piece of art. It's very similar to how you frame up a question, how you approach answering that. So today, Sarah is going to answer a more specific question than yesterday's wonderful general talk. And she's going to discuss the evolution of cryptic coloration in ectoparasites. Let's welcome Sarah.
Thank you all for coming for phase 2. I'm going to turn these front ones back off. Let's see. Let's try that. Okay. Whoa. I don't know how to fix that. It's a little dark. Are you guys okay? Okay, so yesterday, I started talking about my interest in biodiversity. And certainly, a lot of us are interested in biodiversity. The planet is teeming with life. And in fact, if you look at the distribution of biodiversity on this planet, depending on who you talk to, we estimate that there may be as much as 50% of all of life on Earth are parasitic in one form or another. So parasites are arguably the most common lifestyle on this planet. And they're amazingly diverse, and I talked about that a bit yesterday. So today I want to flip the question and ask it in a different way. So in a Hollywood-style way, why is it that there isn't just one parasite species on this planet? Why isn't it that we don't have one parasite that is just exploiting all possible hosts on Earth? What is keeping this Darwinian demon from occurring? Well, we know that parasites are specific, and it's that host specificity that we often attribute to their diversity, the fact that they're locally adapted to some other particular organism. So if host specificity is at the root of whether or not these organisms as parasites can exploit more and more host species, then maybe that's where we should start to look to try to understand the diversity of these organisms as a whole. So you can tackle this question at several different scales. You can tackle it at a microevolutionary scale, looking at ecological and evolutionary processes that influence the evolutionary patterns, macroevolutionary patterns that these organisms experience. And you can look at the macroevolutionary patterns and see if that informs our understanding of how different organisms are related to each other. So in this example, this is a macroevolutionary pattern for this particular group of hosts. This is a phylogeny of pigeons and doves, for example, and this is a phylogeny of parasitic lice that live on these particular host species. And we can compare the phylogenies and look at cases such as these dots where these particular organisms co-speciate over macroevolutionary time. And we can look at these really weird cases where suddenly one species will be found on several different species and look at those as opportunities to study how host specificity can break down in ecological time. And with this information and an understanding about these organisms, we can then conduct experiments to get at the ecological processes that are really driving this pattern of host specificity. We can also look at it at a microevolutionary scale. We can look at the interactions themselves, the behaviors between these organisms, such as this bird that is preening. So birds use their bill to remove ectoparasitic lice from their feathers by preening. And birds spend 10 to 20% of their time every single day preening. This is a major selective force on birds. Time that they spent preening, they can't be used in reproduction, it can't be used in feeding, it can't be used in predator avoidance. This is a major commitment. We can also look at the lice themselves. So these are wing lice on the surface of a wing feather, and these are the barbs along the feather, and these lice will insert their entire bodies in between the feather barbs, such as this one right here, to avoid host defense, because once they're slipped in it, the bird has a really hard time removing them. So we can look at behaviors of both of the players and try to understand how these interactions lead to selection both against the host and selection on the parasites, this coevolutionary dynamic, to better understand this host-parasite interaction. So a lot of what we know about how species interact comes from our understanding of natural selection. And this was for Susanna. These are marine iguanas. These principles are probably best portrayed in some of the early work with comments about natural selection in relation to color. And so both Darwin and Wallace talked about it. Wallace is a little bit more succinct, so I quoted him here. He said, "The survival of the fittest must inevitably weed out those whose colors are prejudicial and preserve those whose colors are a safeguard." And what they're talking about here is cryptic coloration, the evolution of colors that help a prey item avoid a predator. And the evolution of cryptic coloration is probably one of the most famous examples of natural selection that we have. It occurs in grade school textbooks all the way to college textbooks. And you've probably seen this organism before here. This is a peppered moth of the classic Biston-Bechellaria studies from industrial England, arguing about the evolution of cryptic coloration. Those studies have been around since the 1940s, but it wasn't until 2012 that we actually had a rigorous study where there were experimental disclosures looking at the selective forces and the individuals that were selecting against live moths in their natural environment. Not until 2012. This is the best example that we've had in college textbooks. But Crypsis is everywhere. There's a flounder here, there's a Java leaf frog here, and a Java mantid here. We're surrounded by this, so it seems intuitive, which is one reason why it's put forward as such a good example. So I'm going to talk a little bit about the evolution of cryptic coloration today. In order for something to evolve cryptic coloration, three things have to happen. First, there has to be a visually motivated predator. Second, there has to be a prey item, and the prey item has to be on some sort of predictable background. So in the winter, in this habitat, it's very predictable that there's snow, and these prey items are camouflaged against the snow. And that phenotype has to be heritable. Otherwise, even if there's selection, you're not going to get evolution. So let me just zoom in to my favorite part of this interaction right here. This is a golden eagle preening off a feather louse. This is Falcoli purus. And notice that the color is pretty similar to the plumage here. Birds look at their feathers while they're preening, and they can remove these feather lice from their plumage. These birds are obligate ectoparasites, so they spend their entire life on the body of the bird. Feather lice lay their eggs on the feathers, which they glue on with the glandular cement. Those eggs hatch through 3 nymphal instars, again, all on the bird, and then back to the adult, where the life cycle completes itself. They move primarily between individuals through direct contact with mates, or also between parents and the young in the nest. So they are stuck on these birds. They're on a very predictable background, and they have a visually motivated predator. So I look at this as a really great opportunity to study both microevolutionary processes and macroevolutionary patterns in this system. So just to give you an overview of what I'm going to talk about today, we'll look at macroevolutionary patterns in color among species of lice. We'll talk about microevolutionary patterns in color that occur within species, and the processes that are influencing these patterns, which we've been able to get at with experiments. So first of all, has cryptic coloration evolved amongst feather lice? Well, I showed you the one louse on a golden eagle, and that's great, but for you graduate students here, you can't do statistics on a sample size of 1. So to get at this a little bit more rigorously, I decided to use what natural selection has already done, use these natural experiments over macroevolutionary time, and conduct a comparative study where I use sister taxonomic comparisons and compare lice on dark birds and white birds. So this means going through the birds of the world and looking for the most extreme examples that you can find that are closely related. So black swan and a white swan, for example, a brown pelican and a white pelican, right? We've got this black and white theme, dark glossy ibis, which is very dark, white ibis. And we constructed a list of as many of these pairs as possible. Now, you can't just order lice from a white ibis from Fisher Scientific. You have to go get them. So these are the places that I've done field work, and we got lice from birds in all of these places, as well as from colleagues, and then hundreds of years of collecting from people in museums where we were able to go and pull their specimens and look at those specimens as well. So we got lice from as many different pairs of birds as we could. This is what the field site looks like. The collections that we use, so these lice are mounted as microscope slides, especially the historical ones, and we did use these old collections a lot to get the data that we needed. The personal research collection that I've built, as well as the National History, the Natural Museum, the Smithsonian, and other museums around the world, we were able to borrow specimens. So once we went through this list, we were able to get lice from 16 different pairs of white and dark birds. And for each pair of birds, we wanted lice that were in a single genus. So there are different species of lice on these different birds, but they're in the same genus, so we're comparing congeners in this case. Here's the list of birds that we had. So then what we ended up having is lice from a light bird and lice from a dark bird. And we looked at those under the microscope. And under the microscope, we used digital imaging software to highlight the body of the bird, or body of the louse. Sorry, I know that's a louse. And then we controlled for any variation in background color by highlighting a control region and using that as a correction factor. So once we had that, we had a score. Now, lice vary primarily from light to dark. And so just to keep things simple, we just looked at luminosity, which is a measure of how light or dark a louse is. So to get the difference in luminosity, what we did was we took the luminosity of the lice from the light host, and we subtracted the luminosity of the lice from the dark host, and that gave us a difference score, or a contrast score. And here's how the data fall out. So this is a little counterintuitive, I'll explain it. These are our 16 pairs of birds. Each of these represent a light louse and a dark louse pair, or light and dark bird pair. On this axis is the difference in luminosity of the lice. Anything above 0 means that the lice from the white host are lighter than the lice from the dark host. Anything below 0 means it's the reverse pattern. The lice from the dark host are lighter than the lice from the light host. So these are not cryptic, and everything above 0 is cryptic. And in this case, in 16 comparisons, 13 of them all were in the direction of cryptic coloration. And just to give you a feel for what these numbers actually mean in real life, I'm going to show you a picture of one example. And these are the lice on cockatoos. So this is a louse. I showed this last night, but this is Neopsittacinermis louse off of a sulfur-crested cockatoo, photographed on a feather from a sulfur-crested cockatoo. And this is a Neopsittacinermis louse, a different species, but in the same genus, photographed on a feather from a yellow-tailed black cockatoo. And you can see that there's some consistency with the evolution of cryptic coloration in this system. So over macroevolutionary time, we have the pattern is consistent with the evolution of cryptic coloration. But what I've done is I focused all of these data on lice that are found on the body, where birds can preen. And in fact, birds have another group of lice called head lice that are found only on the head. They're specialized for the head. They have a unique morphology that allows them to hang on to the head, but they're not good on the body. They'd get preened off really fast. Birds control these lice by scratching. But this is a little bit of a problem for our assumptions based on cryptic coloration, because birds can't see lice on their own head and they can't preen them off. So I excluded head lice from the previous analysis. But instead, we did the same comparison just with head lice. So we found birds that had dark heads, birds that had light heads, and we looked at their head lice. And in this case, we were able to find 10 comparisons of head lice on light and dark birds. And here's what happens. So this is the same pattern as before. Anything above the line is cryptic. Anything below the line is conspicuous. And in this case, out of 10 comparisons, 6 of them are conspicuously colored. They're not cryptic at all. So there is no evidence for cryptic coloration in head lice. And in fact, if you look at head lice, this is what they look like. So even on very lightly colored birds, you get these very dark lice. So overall, head lice seem to be pretty dark. So what I've shown you so far is that typical feather lice, so these are the lice on the body of the bird, they have evolved cryptic coloration among bird species. And head lice haven't, but you expect that. So really they're the exception that proves the rule in this case, because they're the lice that the birds can't see and they can't preen. So I'd like to move to another example now. This is lice on gulls. So gulls vary in color from light to dark. And they're really interesting because they're parasitized by a single species of louse. Now, most species of lice are really specific. They're only found on 1 or maybe 2 host species. But this louse is found on 27 different gull species. And look at the different colors of these gulls. How is this possible, right? If there is evolution of cryptic coloration, this doesn't make any sense. Well, if you look at the specimen collected from different hosts at different geographical areas, you see that the lice can be dark like this, or this species can also be very light like this. So this is intraspecific variation in color. And this variation was described in the 1950s by a taxonomist who actually described subspecies of lice. Most people don't describe lice to subspecies, but he noticed this variation in color, and he described them as different subspecies solely based on these pigmentation patterns, from very dark to very light. And he actually described 8 different subspecies. So what we did is we looked at the distribution of these subspecies, and then we scored the color of the hosts that they're found on. So here are the data of this. The lice go from dark colored to light colored up here based on what rank subspecies they are in terms of color. And then we scored the color of the birds from dark. This is a lava gull. And there are several species of dark gulls, which is why this point has error bars. 27 species of gulls are represented here. To light, like this Iceland gull, which only has one host that has this particular subspecies. What we see is that there's a strong linear relationship between louse color and host color. So within this system, there's some sort of sorting event that's causing these lice to be distributed only on the color hosts that would match their color pattern. Now this is really interesting. It could be that there is convergent evolution of color morphs, because these birds, these dark birds, aren't closely related. So it doesn't make any sense that a bird that's in the Galapagos and a bird that's in the Caribbean or in South Africa might have the same subspecies of lice, because they're not even closely related. So it could be convergent evolution of these different lice. It could be massive dispersal and sorting events, and it would be really interesting to get at that. I tried to use some citizen science approaches to get gull samples, but it doesn't work so well. But there are other species that have variable lice that are a little bit more easy to work with. See these guys over here? They're nice, calm birds. So I work on pigeons. Pigeons are really calm in captivity, and they're also a very diverse group of organisms. There's 300 species of pigeons worldwide. They're found in most habitats. They're diverse in color. They're diverse in size. They're actually really beautiful birds. And they're also really common in the cities, which makes some experiments really easy to do. So I focused on columbiforms. So this is a lemon dove. And what I've told you so far is that lice vary predominantly in color from light to dark. Well, that's great, and that makes some intuitive sense. But what about being camouflaged on something that's brightly colored? Right? How does that work? The lice are never yellow. So I'm going to conduct an experiment right now with you guys. And I want to show how lice might be cryptic on this color host, even though they can only vary in one mode of color. So close your eyes and think about the color of this bird. Just close your eyes for a minute. And when I change the slide, I want you to look up, and there's going to be 3 different lice on the screen. And I want you to think about which one you see first. Okay? Open your eyes. These lice vary only in one color, that is black to white. They only vary in their luminosity. So how many people saw the white one first? How many people saw the black one first? How many people saw the gray one first? See, there's some people that see gray, but you guys are a minority. And this doesn't have any orange, it doesn't have any yellow in it. So it's like that joke with the polar bear, and there's two guys that are tying their shoes on, and the guy says, well, you can't outrun the polar bear, don't put your tennis shoes on, right? He says, I don't have to outrun the polar bear, I just have to outrun you, right? That's how selection works. This guy doesn't have to outrun the polar bear, it just has to outrun these guys. And so that's how you can get selection, even in a group that doesn't vary in different color, except for in luminosity. So I just wanted to put that out there because birds are very different colors, yet this applies even in systems where the birds aren't just black and white. So here's the species I just showed you, and then just to show you again how this works on a dark colored host. This is Columba arquatrix, a Ramaran pigeon from South Africa, and this louse is much more cryptically colored than either of these two. All three of these lice are the same color in this version and in this version. They're not varied, it's just that these two jump out a lot more on this particular background. So the first thing I wanted to do before I embraced pigeons as a system for studying cryptic coloration is I wanted to know if cryptic coloration evolved amongst the lice in this system in nature. Each of these represent a different genus of pigeon, and I looked at the darkest pigeon and the lightest pigeon I could find in each of these genera. And again, I compared luminosity, and anything above the 0 line means they're cryptically colored in that genus based on the same data I was showing you earlier. And in all 6 cases, the lice on the lightest pigeon is the lightest colored louse. And in this case, we have birds that are quite light, like the Ducula bicolor, which is an imperial pigeon in the South Pacific. It has a very lightly colored louse, Columbicula wolfugulae. And this is the city pigeon, which has a relatively dark colored louse, right here, Columbicula columbae. So across many species, again, we have this pattern that amongst the different species of lice, we get evolution of cryptic coloration. These species of lice are about 25 to 30 million years divergent, just to give you an idea of how long ago they diverged. So this is a good system because we see that the pattern exists in macroevolutionary time. Now, what can we do with it experimentally? Well, I want to look at it within a species because I can manipulate that. And pigeons are especially good at that because even within a flock, we can see different birds that are very different colors. Thanks to all the crazy domesticated pigeon breeders out there, we have a lot of domestic genes that make it into the flock and make it extremely manipulatable. So first we have to catch them. This is my graduate student right here, Emily, who's trying to catch pigeons. And this bow net works pretty well, but it turns out that city pigeons are pretty hard to catch, partly because they're overfed. Everybody feeds them. And so they're a little bit wary of the bow net. So next she tried putting traps underneath overpasses, and those work pretty well until people come and take the traps. The cops like to figure out what you're doing when you're putting traps underneath the overpasses. So she's had that problem a little bit. But one of our favorite methods of catching birds is this one. We just use it to stun them a little bit. No, we don't do that. But we catch the birds mostly with traps, and we bring them back to our facility on the roof, our pigeon lofts on the roof of our biology building, where we're able to, Utah's really great for this. You can put dehumidifiers in the shed and drop the humidity extremely low. So in Utah, we can get the humidity, well, you guys know it's low here, but we can get it to less than 20%. I think we're the only people in the state that buy dehumidifiers, right? But we put them in the sheds and we can get it really low. And what happens is all these feather lice get all of the moisture that they need from the air. They actually gulp air and use the water droplets that they gulp from the air. When the humidity is really low, they can't do that. And over a period of about 8 to 10 weeks, you can totally eradicate all of the lice on a bird. So without using any chemicals, we can eradicate any naturally occurring lice on the birds, and then we have clean birds for experiments, louse-free birds for experiments. So we do that. We catch the birds, bring them in, get rid of their lice, and then we can put lice on them. So we have, inside the sheds, they're individually caged so that we can manipulate lice individually on birds. We catch some black birds and some white birds and with everything else. How we move lice around, we have birds that have a lot of lice, and we put them in these fumigation chambers. Thanks to Costco and popcorn, they make big jars of things. And we pump CO2 into these chambers, and it anesthetizes the lice, and the birds are fine. Pigeons are really calm, and they're great this way. And then you ruffle the pigeon over a tray, and all the lice fall off. And the lice wake up in about 5 minutes, so you can move live lice from one bird and put them on another bird in an experiment. So it's great in that you can manipulate it that way. So the first thing we did was we anesthetized the lice off of the birds that we caught in the field. And some of the birds were white, and some of them were dark. So we just wanted to look and see what happened in the variation in these different birds naturally that we see. Well, on white pigeons, so let me actually orient you. So we caught gray pigeons. That's the natural wild type color of a rock pigeon. Then there were white pigeons that we caught from an area where they were escaped homers, so they'd been isolated for a very long time. There were only white birds in this particular farm area. And we looked at the luminosity of the lice. So the luminosity goes from dark to light. This is what the gray pigeons were, and you'll see this dotted line on a lot of slides. That's what you expect from a normal gray pigeon that can preen normally. That's the luminosity. It's all relative. And the white pigeon had much lighter lice, significantly lighter lice on these particular birds. Now, this wasn't an experiment. This was just what we noticed in birds that had been isolated for some unknown amount of time. So it's certainly consistent with a difference in color between these birds. But one question that we had is, these lice are eating the feathers. These feathers are different in color. So how do we know that it's just a change in color in the lice, or is it just the diet that is being sequestered in their cuticle? So we decided to test whether or not lice are what they eat. And we put lice on live, newly hatched baby lice in the incubator on feathers that were white and on feathers that were black. Put a whole bunch of lice in on tropical, warm, moist conditions, their favorite conditions in the incubator. And we waited for a month, which is enough time for a newly emerged louse to make it all the way to adulthood, which is their last molt, and that's going to be their final color. And then we scored the color of these lice raised on these different feathers. And we also know that the lice eat the feathers, because I was talking with people at lunch about poop and how you study it. Well, you hit the bottom of the barrel when you're studying louse poop. So here's pictures of louse poop here, and we know that the lice are eating these feathers because we're getting the frass at the bottom of the vials. So we know they're eating it, but yet when we look at luminosity, there's absolutely no significant difference in the color of the louse. If you're going to read anything into it, the lice on the dark feathers are actually slightly lighter, but again, there's no difference there. So diet is not influencing louse color in this system. This also makes sense because remember I showed you the head lice. The head lice on white birds are dark, and if it was just the feathers that they were eating, the head lice on white birds shouldn't have been dark when we were looking at the macroevolutionary comparisons with albatross. Just to make your head itch a little bit, here's some pictures of human lice. Human lice also vary in color from light to dark. And we haven't been able to breed feather lice in the incubator long enough to do any kind of real breeding experiments and get at the heritability of color yet. We're working on it. But we do know that color is heritable in lice, because there was this work done in the 1940s by this guy named Busbein who took lice, human body lice, and scored them from light to dark and did sibling crosses, where he took parents that were dark and he scored them based on these 4 different categories each generation, and then crossed them with parents of known colors. And by the time he got to 4 generations, he could take lice that were all dark and he had very dark offspring, lice that were from light parents, and he had almost all white offspring. And if he crossed them with dark and light parents, he got a more mixed category of more morphological variation at that point in time. So we know that there's heritability in louse color from these studies, so there's no reason to think that feather lice are any different. So thinking about the categories of data that you need to demonstrate the evolution of cryptic coloration, we know that they're on a reliable background. Lice are on a reliable background. We know that there's heritability, and now I want to talk about selection. So how do we get different colors of lice? Well, pigeons are great because they already come in different colors in the city park, but to move into some urban, more urban ecology, we use fingernail polish to create light and dark lice. So this is a project for a very talented undergrad who likes to paint with a single hairbrush. And she painted several thousand lice for us, and we recreated very light lice and very dark lice that we could put on either dark feathers or light feathers. So once we had this phenotypic variation established, we put these lice either on white pigeons or on dark pigeons. We painted almost 500 different lice, and we divided them so that each bird got 30 white lice and 30 dark lice. So each bird got 30 conspicuous lice and 30 cryptic lice. And the colors differed depending on what color the bird was. So on the black bird, the black lice are the cryptic lice, and the white lice are the conspicuous lice. There were 8 pigeons in each treatment, and we waited for 48 hours. So the birds were put back in their cages, and they were allowed to preen. And after 48 hours, we CO2'd all of the lice on the bird and recovered all of the lice, and then we counted up which category they were in. So here's the data. These are the number of lice that were recovered off of the birds after 48 hours, and they were either cryptic or conspicuous. Significantly more of the conspicuous lice were removed from the birds than the cryptic lice. So this is consistent with selection against conspicuously colored organisms. But we were able to manipulate one more thing to really get at it just a little bit more, and that is we can create basically a behavioral knockout by putting a C-shaped plastic bit between the upper and lower mandibles of the bird. This doesn't affect their ability to feed. They feed just fine. In fact, they're really fat. But it prevents them from touching the upper mandible to the lower mandible. And by doing that, they lose that forceps-like action that they need to actually remove lice from the plumage. So by doing this, they can no longer remove lice. They still preen. They still go through the motions of preening. They think they're probably effective. They still preen just as much, but they can't remove the lice. So we repeated this exact same experiment and put painted lice on white and dark pigeons that couldn't preen. And this time, here's the data that we get. The number of cryptic lice and the number of conspicuous lice that are found on the birds after 48 hours are exactly the same. Actually, one might be 97 and one might be 98. I can't remember. But the p-value is 1. There's no difference between this. So this shows experimentally that birds are the selective agent against conspicuously colored lice, and it's preening that is causing it. We know that this is the interaction. Yeah, preening selectively removes conspicuously colored lice. One more experiment. So we know that there's selection. What we really want to know is, do lice change in time if they're put on different colored hosts? So if you take a population of lice that exhibits natural variation, and there is some variation in color within a natural louse population, if you put it on different colored birds, can we watch evolution in action and see a change towards more cryptically colored organisms? So we ran an experiment where we transferred lice from gray pigeons, the wild type color, to gray pigeon controls and to white pigeons that could preen normally, in this case, and to birds that were bitted, in this case, so no preening in this case. We put 75 lice on each bird. There were 6 birds per treatment. And then we waited for 9 months. This is about 10 louse generations. So here is the score in louse luminosity at the end of the experiment. Again, everything's set at 0 based on a normally preening gray pigeon. And what we found is that birds that couldn't preen are not significantly different than this control. They don't vary in color. But lice on the white bird that can preen are significantly lighter than lice on birds than the control, which is the only category in this whole experiment that you would expect to change if it's preening-mediated selection for cryptic coloration. So the number of units that are different here, there's a change in about 4 luminosity units. So just to put it in perspective, the lice that I showed you over the course of macroevolutionary time on the white pigeon from the South Pacific and on rock pigeons that diverged 30 million years ago, those differ in luminosity units by about 80. So these are significantly different, but much less so, but in the direction that we would expect. This is only 10 generations. This isn't 30 million years. This is 10 generations. So we have a relationship at this very proximal microevolutionary level showing that we can demonstrate evolution of cryptic coloration in this system. Does this happen in other species? It's not just lice that are ectoparasites, right? There are 70,000 species of ectoparasites that we know about, and they're not unique to birds. So these are parasitic crustaceans. Most of these are the same species. So there's some other interesting parasites out there that need to be studied. They're a really powerful system for studying between this micro and macroevolutionary scale. So I'd just like to summarize briefly what I've talked about today. First, we talked about cryptic coloration, and that has evolved over macroevolutionary time, which is true on many different families of birds of the world. We looked at head lice, and we found that they're not cryptically colored, in part because they violate several assumptions of what would be expected for cryptic coloration, and in that way, they're an exception that proves the rule. There's evidence that cryptic coloration occurs within a parasite species, such as the species that's found on gulls, Quadriceps punctatus. And we know from experiments that birds selectively remove conspicuous lice by preening, so we have the selective agent. The data that we've got from these multi-generation experiments are consistent with rapid evolution of cryptic coloration. What we're going to do next is look at the variation that is caused by these experiments. We have some experiments that are running now that are going to be running for 3 years, and we're sampling lice as we go to do genomics, and hopefully we'll pick up some signatures of color change. But what we're doing is in these different lineages, we like to isolate genes that change and then compare them to the macroevolutionary patterns of the different species of lice that are found on pigeons and doves of the world, and see if we're getting the same changes in our experiments as we see using phylogenomic approaches across all of these species of lice found on pigeons and doves elsewhere that vary normally. So we're bridging into the genomic era. I don't have any data on that yet because we're just getting the reference genome assembled at this point, but we're getting there. We're starting to get to the point where we can get a genome out of a single louse, so that'll be good. I think this is an exciting way forward to bridge a lot of these concepts. A lot of this work is summarized in a book that's going to be coming out with my collaborators in crime, Dale Clayton and Kevin Johnson. The 3 of us have been working on kind of a summary of the system. So if you're interested, it'll be coming out in the next probably 6 months. We're excited about that. Obviously, this work isn't possible without an army of people helping, and this is my lab with the army of graduate students and undergraduates that are involved in all this research. And with that, I'd be happy to take any questions. Yeah?
What about lice that specialize on birds that are highly sexually dimorphic? With plumage color is very different.
Yeah, I've been interested in looking at that, but I don't have enough data to know whether or not there is actually a difference in either where the lice partition themselves. We know that in some cases males have more lice and females have more lice depending on the system. So I don't have enough data to really answer that. I would think that something might be going on, but I just don't know. Yeah.
Kind of along the same lines, one of the criteria you mentioned is that the background is used. So what do you think happens in birds where they vary plumage across different seasons? Like if their plumage changes dramatically during the breeding season, do lice sort of crash on those birds? Like they put them all off? Or
Well,
do birds have fewer lice in general?
it could be that there may be fewer lice in different seasons. We don't know. Lice often have cycles where they have high and low loads, and it may correlate with different plumages. There's also birds like magpies that have really dark patches and really light patches. And we know that lice are photosensitive, and there have been some studies on barn swallows where they've looked at lice and they find that lice might specialize on the white spot instead of the dark spots. So it's possible that they're also sensing the color of the bird and going into a microhabitat that would be consistent with their color. I want to do experiments where we get pigeons and use hair dye and put like polka dots on them and see if we can detect any kind of microhabitat selection. But we haven't really been able to get it out. But we do know that lice are microhabitat specialists, and there just isn't enough data to really figure out where the lice are specializing on a lot of these birds. And part of it is in order to collect the lice, a lot of the times people have killed the birds. And as soon as you kill the birds, their temperature drops, and lice, for some reason we don't know, go to the head of the birds. And so by altering the host, you alter the microhabitat use of the lice, and therefore you can't get the microhabitat data. And so there are ways around it, but it involves a lot of live birds and a lot of really tedious work trying to census things on birds that are flopping. But I'd like to know. Yeah?
So I'm interested in pigeons in particular. It seems to me that I've seen pigeons allobreed. So you'll get two pigeons next to one another grooming each other.
Yeah.
And so I would have thought that would then prevent the pattern that you found, where you found no criticism.
Yeah, no, so a lot of birds allogroom, and there's been no evidence that allogrooming actually helps remove ectoparasites. Allogrooming is typically most commonly focused around the head, and so if we thought allogrooming was going to be especially effective, then we would also see some pattern of crypsis in head lice, and we don't see that at all. There's some evidence that allopreening helps remove ticks in penguins, but that's the only thing that I've noticed that there's actually been data on that. It seems that allogrooming is more a behavior to reinforce pair bonds than actually something that's very effective at grooming ectoparasites. Yeah.
So pictures of fish you showed with parasites on them, parasites were mostly cryptically colored.
Yeah.
Do you have a hypothesis, I guess, for why that would be? Since I don't really think do fish groom each other?
This is a really good question. So how is this the case? Well, there are cleaner fish where fish groom other fish. I think that in this system, these crustaceans, which are ectoparasites, may actually be undergoing, instead of cryptic coloration, to be removed by cleaner fish, which could happen, nobody knows. But it also could be cryptic coloration to avoid having their host eaten. If they are a different color and then it makes their host more conspicuous, then they get eaten. And these parasites don't live inside another host. This is their final host. So if they get eaten, they're dead too. So they might as well help their host be cryptic colored. Selection should work to maintain crypsis on the host as a whole. But these parasites are really cool and nobody's studied them. But there's also evidence of variation in color in ticks. Nobody's looked at that. There's fleas that vary in color a lot. Parasites on mammals would be really interesting because there's nocturnal, but you have some diurnal mammals, and those would be interesting because you might expect cryptic coloration in those parasites, but not on the ones that are nocturnal, depending on when they groom. Yeah?
We have some animals that operate in the night. Their eyes are real big.
Yeah.
I'm wondering, and then if they operate in the day, they're not as big. So I'm wondering, I don't know if they have eyes and lice, but whatever it is that they use to see color, Does it look different regardless, you know, if they're operating on a dark species versus a...
Are the eyes on the lice different?
Yeah, in other words, how either they're bigger or how they receive the light in some way, you can tell the difference.
You know, that's a good question. There are some species of lice where we don't think they have eyes at all. But the eyes seem pretty rudimentary compared to other insects. We know that they can sense light, though, because if you shine light at them, they run away. They're negatively phototactic. And we use that in assays occasionally when we want to get them to move. You can flash light at them and make them run. So they can certainly sense it, and I would think that if they're in a feather that's darkly colored and plumage that's dark, less light would be transmitting, and they would respond to that in some way. So all the lice we've worked with at least can sense light.
You try different color lights, I think.
I haven't. I don't know anything about color vision in insects, especially this group, but that would be interesting. Yeah?
Do the birds equalize? Do they gain nutritional value?
Well, they're only eating the bird's feathers anyway, so these aren't very nutritionally advantageous, but you could argue maybe they have some sort of endobacteria that's providing some essential amino acid or something. So we've actually taken birds and force-fed them lice and then calibrated how many lice they actually eat by looking at what's in their guts. And I think they probably eat about 5% of the lice that they preen. These are on extremely heavily loaded birds. And so that suggests to me that those are just kind of incidentally ingested, that they're not actually eating them as a means of getting any kind of nutritional value. They mostly just crush them up and spit them out. Well, thank you all for coming. I'm happy to answer any questions if you want to come up afterwards. Thank you.